Welcome back. This is the second lecture in chapter 9, and we're going to start back here with the diagram that we ended the first lecture with. And so we were talking about, we've got these 4 different types of possible offspring that inherit from these different gametes. So we've got the possibilities of white genes, white flower genes, purple flower genes, all coming from the adults. And so this brings us to what Mendel's law of segregation actually is saying. So some of this already makes sense from what we know from things like meiosis. Now remember though, Mendel didn't know about things like meiosis necessarily because we hadn't really looked at that yet. Also, he didn't know the term gene, and so you'll notice that some of those words don't actually show up here in this idea. And so this is Mendel's law of segregation, these ideas to explain this inheritance pattern. So one is that each parent is going to put into every sperm or egg, depending which one they're producing, a single set of instructions, aka a gene, for building that trait. And so this would be, for example, the flower color. So be the directions to make the purple flowers or the white flowers. So they get one set of directions in their sperm or egg for that trait. Second is that the offspring therefore find themselves with 2 copies of the instruction for that trait, or they find themselves with 2 copies of the gene for the trait. We know them as alleles, right? Remember, allele is the different versions for any trait. So in this case, we know the alleles are purple flowers or white flowers. The actual trait produced by an individual depends on which 2 copies of that gene, of the directions, they inherit from their parents. And so this brings us to some new terms. We saw these, you may have noticed them on the last diagram. I told you we'd come back to them. Homozygous and heterozygous. So you've probably heard homo, that prefix, before. So homo means what? It means similar. So homo means that the 2 copies are the same, and hetero, that's right, means that they are different from each other. So let's go back and look at that diagram again. And if we'd look at the top, we started with heterozygous pea plant parents, meaning that the genes for this trait were different from each other. We had a white flower gene and a purple flower gene. We see that each of the gametes gets 1 copy from the parents, just like we saw in Mendel's law there, and those genes go into the offspring get 2 copies, and those 2 copies are going to dictate what they get as far as the characteristic. And so you can see down here, these have been labeled with homozygous or heterozygous. So you can see the 2 here in the center are heterozygous, just like the parents, 1 copy of each. We've got on this side homozygous, homo for same, so they've got the same copy. And these are both the purple gene, which we discovered in our last lecture for pea plants. Purple is dominant, meaning that it is going to mask or cover up the other gene, in this case the white. So this is going to be homozygous dominant because it has both copies as the dominant gene. And then on the other side, we've got homozygous, again, because it's the same copy, both white flower genes in this case, and it's homozygous recessive. So you can see from the picture here, homozygous recessive, those copies determine what the trait will be. 2 copies of the white gene, we get white flowers. one copy of each, a copy of the dominant, copy of the recessive. The dominant is going to win, if you want to think about it as win, and it covers up the white gene, and we're going to get purple flowers. And then if you have nothing but purple genes, obviously your flowers are going to be purple. And so what this means too about those true breeding flowers, the flowers that Mendel started with, those true breeding flowers were homozygous for each of their genes. Homozygous dominant for the purple, homozygous recessive for the white. All right. So the next thing I want to look at is looking at observing an individual's phenotype is not enough to determine its genotype. Now, I think we talked about these words a while back, but just to remind you, so here, looking at this picture, this gives us an idea of the phenotype and the genotype. So phenotype for this giraffe here, that's the one that we're looking at. So the phenotype for this giraffe is that it has little or no pigment in its eyes, its hair, and its skin. The genotype is that it's homozygous for the recessive allele for albinism. So it is an albino giraffe. They are rare, like a lot of albino animals are in the wild. So let's take a look at, again, the definitions for phenotype and genotype. You might be able to guess them from just looking at what we've got here. So phenotype is the outward appearance of an individual. So this is the physical characteristic. Okay, so it's what we can see, what we can observe about the organism. So that's the phenotype. Underneath that phenotype is the genotype. The genotype is actually what is creating the phenotype. So the genotype is the genetic part. And so that's how I remembered it. Genotype sounds like genes or genetic. Phenotype sounds like physical. So phenotype is what you can see, the physical part. Genotype is the genetic part, the part that's coding for the physical part of the organism. Okay, and so with our giraffes there, the physical part is, in this case, we're looking at coat color and eye color. The genotype is telling us what genes are actually coding for that phenotype, for their coat color. Okay, so with the albino giraffe, we've got our albinism, we've got recessive genes. It's homozygous for those recessive genes to get that albino coat color. So we've got our homozygous dominant, heterozygous, or homozygous recessive, the ones we were just talking about. So, sorry, if we go back to our giraffe here, we know that this one is homozygous recessive. The question is, can we tell what the genotype is for these giraffes? We know that the phenotype is they have pigment for their hair and skin. We can tell that. We have to assume that they have pigment in their eyes as well. So we can tell that they do have dominant genes. But can we tell if they are homozygous dominant or heterozygous? Because we know that this giraffe up here is homozygous recessive for albinism. But can we tell if these giraffes back here carry the recessive gene for albinism? And the answer is no. We cannot, from observing phenotype, always tell the genotype. We can tell the genotype if you're showing the recessive condition, but we cannot tell your genotype if you are showing the dominant condition, because if you're showing the dominant condition, you can be homozygous dominant or heterozygous if you have the dominant phenotype for whatever trait we're looking at. So how can we predict what, if those are homozygous dominant, heterozygous, those giraffes out there, or if we know what they are, say we know that one of those giraffes is heterozygous, if we want to breed giraffes together, how do we figure out what they might have as far as offspring? That's when we're going to use Punnett squares. You may have heard of these, you may have done them in high school. And so that's what we're going to look at next, is we're going to look at how to do Punnett squares. I'm actually going to start a new video on how to do Punnett squares and probabilities for analyzing these so that we've got plenty of time. So I'll pick up in the next video with Punnett squares.